My name is Hallie Harima, and I am a second year studying biology at the University of Cincinnati. Today, I will be presenting on the development of constructive taste bud expansion across Astyanax mexicanus morphotypes. As an outline of my project, I'll be going over the background, overview, methods, and results. And to start, I'll go over the background. The fish I'm studying are the Astyanax mexicanus. They're a Mexican tetra, located in the freshwater region of the Sierra de Abra in Mexico, as you can see outlined in red below. There are two distinct morphotypes, the surface fish and the cave fish. The image on the right shows the surface fish, which looks like any old fish you'd see in a river, whereas here is a cave fish. The cave fish lives in an extreme environment where there is no sunlight and has lost its pigmentation and eyesight over time. This is what allows the cave fish to be an ideal candidate for a study, which the focus of my study is on the taste bud. They are sensory organs. They have hair cells in a bulb, which contains receptor, structural, and basal cells. The papillae allow the chemical messages to be sent to the brain. So the food particle lands on the receptor protein, which sends a nerve impulse to the brain that says, "This is what I'm tasting," which there are 5 tastes: bitter, sweet, sour, salty, and umami. Have extra oral taste expansion been seen in other fish species? The answer is yes. Catfish live at the bottom of lakes where it's very dark and hard to see, similar to the cave fish. The barbels help catfish be able to taste. As the barbels increase in size, the number of taste buds increase as well, showing an expansion. For the overview of my project, different numbers and locations of extraoral taste buds have been seen between the two morphotypes. The purpose is to pinpoint the timing of positional expansion and identify possible genes involved down the line. Here's a diagram of the extraoral taste buds in the fish. On the left is the surface fish, and on the right is the cave fish. As you can see, the cave fish has a lot more taste buds than the surface fish, whereas the surface fish just has them around the lips. The method of my experiment. There will be surface and cave fish taken on monthly developmental intervals between 6 months and a year and a half. I'll be using fluorescent histological stain to mark the taste buds, which here on the right is a taste bud cell. I will first stain with an anti-calretinin antibody, the primary antibody. The anti-calretinin is a calcium-binding protein. It binds to the calcium specifically in taste bud hair cells and marks them, which the secondary body is specific for the anti-calretinin, which attaches. The secondary antibody has fluorophores, which are visualized with specific light wavelengths on a stereoscope, where under that light, the fluorophore will beam red, showing a taste bud. Here are the results of my experiment. At 6 months, there's no difference of taste bud expansion. At 9 months, there's an intermediate stage of cave fish taste bud expansion. Here's a dorsal view of the adult fish. On the left here is a surface fish, and on the right is a cave fish. As you can see, right here, the extraoral taste buds have expanded on the cave fish, whereas on the left, on the surface fish, there is no expansion in the same region. Here's a side view of the adult fish as well. As you can see right here, on the lips of the surface fish, there are taste buds, whereas here on the right, on the cave fish, there's been taste buds that have expanded down its face. Here's a 6-month-old cave fish. Here's the dorsal view, and there is no extraoral taste bud expansion. Right here is where we expect to see taste buds, but there are none that have developed yet. Here's a 9-month-old cave fish. On the dorsal view here outlined, you can see the taste buds have begun to expand. And here's the side view where the taste buds have also begun to expand on the 9-month-old cave fish. In my future work, I will continue staining for each stage of development. I will quantify taste bud numbers and analyze extraoral taste bud location using ImageJ to find statistically when the expansion is significant developmentally. Then, I hope to find genes responsible for expansion using QTL analysis. Thank you for watching my presentation.